My name's Joyce Harper and I work at University College London. I've been working in the IVF field since 1987, and then in 1992 I joined the Hammersmith team to work on pre-implantation genetic diagnosis. I do a lot of work within ESHRA and also the Human Fertilisation and Embryology Authority, and I've been collaborating with Genesis Athens for many years. In this short video, I'd just like to give you an overview of where we are with pre-implantation genetic diagnosis and pre-implantation genetic screening. Just one disclosure, I've just this year in 2014 started a company called the Embryology and PGD Academy, and we deliver hands-on training in embryology and PGD technology. And through this talk, I'll give you a few references of papers that you can go to read up on. Here's one on PGD state-of-the-art from 2011, written by myself and Siobhan Sen-Gupta. And when I was chair of the ESHRA PGD Consortium with Gary Harton, we wrote a paper on the use of arrays in PGD and PGS. So what I'm going to talk about today is the difference between these two procedures. I'm sure you've heard of them before, but people do get confused about the difference between PGD and PGS. The methods that we use, which is a very dynamic area at the moment, there's lots of new technology that's come in. And as I mentioned, the ESHRA PGD Consortium, it was a consortium we set up in 1987, and I'll be talking to you about some of the data from there. And to finish off, unfortunately I have to mention misdiagnosis in PGD and PGS, and then the disadvantages and advantages, the ethical dilemmas, and the future. So what is PGD? Now, it may seem obvious, but I just really need to set the scene. So for PGD, we're selecting genetically normal or carrier embryos or chromosomally balanced embryos from patients who are at very high risk of transmitting this to their child. So they come to us knowing that they're carrying this abnormality, and we can do the genetic test for them. So the aim is to transfer an embryo that will develop into a healthy child. So we want to do a diagnosis for the specific abnormality they're carrying. So most of these patients will actually be fertile. They could have got pregnant naturally and gone through prenatal diagnosis, but PGD is another reproductive option for them. The diagnosis we do is specific at the moment. There's new technology that's coming in, I'll mention at the end, but at the moment we do a specific test for that abnormality. And in this situation, because these couples are at high risk, any undiagnosed embryos must never be transferred. There's always a risk that an undiagnosed embryo may be affected. Ethics and controversy, well, we can do PGD for almost anything, and we can do it for late onset disorders and also traits and characteristics. So for example, social sexing, because the couple just want a child of a particular sex, has been done for many years, but in the EU it's actually illegal to do this. So I'm sorry you can't read this last bit, but it just says there's multiple approaches that will lead to future ethical issues in here. So what sort of patients opt for PGD? Well, they may have an objection to termination of pregnancy. They may be infertile but carrying a genetic abnormality. So if they're infertile and they're having to go through IVF, it's very sensible to add on the PGD step. But if they're fertile, they may want to try other ways of getting pregnant. They may have had repeated terminations because they've got pregnant, had a prenatal diagnosis, found the embryos to be affected, or the fetus to be affected, and then they've terminated the pregnancy. So they may have had repeated terminations and just feel that they've got to try another reproductive option. They may have had repeated miscarriages due to a chromosome abnormality. And there's something called saviour sibling, which I'll come back to more at the end. But this is where the couple have an existing child who's ill from a very lethal condition, such as some thalassemias, etc. And they have the potential of saving that child's life by doing HLA typing. So what we would do is they'd go through PGD, we'd check, obviously, if there was a genetic abnormality, we'd check for that as well, but we genetically match the embryos that were generated from the IVF to the existing child with the aim of, at delivery, using the stem cells from that matched child to cure the existing child and hopefully saving their life. In the UK, we're governed by the Human Fertilisation and Embryology Authority. They have a lot of information about PGD, and they also have a list of conditions that are licensed for PGD in the UK. So you might want to go and visit that website and have a look at that information. So that's straightforward, but what about PGS? What's the difference between PGD and PGS? Well, maybe in the future there may be more of a blurred line, but at the moment PGS is basically an embryo selection mechanism. It's using the same technology that we have in PGD, but to help infertile couples going through IVF to select embryos according to their chromosomes. So we're selecting the normal diploid embryo, euploid embryo or oocyte, trying to improve the IVF delivery rates. So these are couples, as I said, going through IVF. So what we want to do really is diagnose as many chromosomes as possible. Some very early methods using the FISH technique, which I'll come back to, just looked at a few chromosomes, but now we have methods where we can look at all of the chromosomes. The ethics of this, if we're choosing a new embryo selection mechanism, we should have evidence to show that this procedure has actually worked and that using this procedure will give us a significantly improvement in delivery rates in couples that are going through this compared to a control group who's not gone through the treatment. And at the moment, the randomised controlled trials have not proved any efficiency. Up to recent years, there was 11 randomised controlled trials showing that actually some of them showed a significant negative effect of doing this procedure. And there's been a few recently using some of the new technology, but they're again quite new papers and there's various issues within there. But we hope in the future there'll be more papers that come out, more randomised controlled trials showing us that this procedure is viable. So at the moment, my view is that the best approach is still debated. So the indications for PGS, obviously advanced maternal age, that was always be the sensible one. We know that because of meiosis problems, as a woman ages, the chromosomes in her eggs can become very unstable, and certainly past the age of 35 have a huge effect on fertility and on miscarriage. So looking for chromosomes in embryos from these patients might be a very useful thing to do. The trouble is that these patients produce fewer embryos, and so that leads to problems with embryo selection. If a patient has recurrent IVF failure, well, there may be many reasons they have recurrent IVF failure. It may be a chromosomal reason in their embryos, so we could test it using PGS. If they have recurrent miscarriage, again, many reasons for recurrent miscarriage, but if the karyotypes are normal in the parents, there may be other immunological reasons, or it may be something to do with the chromosomes in the embryos. Severe male factor, and at the bottom here, I've written everybody. Maybe in the future, this is a technique that everyone going through IVF will actually have. So the stages of PGD and PGS, you probably know these, but we've got to obviously generate the embryos in our IVF lab first, and then we're going to do embryo biopsy, where we remove some of the embryo, and then we take those cells that we biopsy, and we'll do our genetic test on those few cells. So what we would do on a PGD case, we would biopsy all of the embryos that got to the right stage, and then take those cells and do our genetic analysis, and then from those results choose which embryos would be best to transfer. Now in future videos like this, hopefully we'll have some videos of showing you how we do embryo biopsy, but in this talk today I'm just trying to give you an overview, so I'm just going to quickly mention we can take the first, and definitely we need the second polar body from the oocyte to do our genetic tests, but this will only tell us about the chromosomes or the genes in the mother. They won't tell us about any father's genes. We can do cleavage stage biopsy, where we take one cell, one blastomere from the cleavage stage, and this was done for many years, and this was the original technique that we used to do at the Hammersmith Hospital. And now in recent years, blasters biopsies become very popular. And Georgia Kakali from Genesis Athens has done a lot of work on this and published some of the groundbreaking papers that shows that we can take some of the trovectoderm cells, leaving the innate cell mass intact, and use these trovectoderm cells for our diagnosis. So I just wanted to mention mosaicism, and this has been one of our problems with PGD and PGS. When we're doing embryo biopsy or biopsy of any tissue, we need to have that tissue representative of the sample that we've taken it from. And unfortunately, because of mosaicism, chromosomal mosaicism within human embryos, this is not the case. So it's present at all stages of pre-implantation development. Work by myself and Joy Delahanty, Santiago Monet and others showed more than 20 years ago that up to half of cleavage stage embryos are chromosomally mosaic. And there's more recent evidence now, lots of people are looking at blastocysts to show that at least about a third of blastocysts have chromosomal mosaicism. So again, I haven't got time to go into all of this now, but it is problematic for PGD and PGS in some circumstances, especially when we're looking at chromosomes, because the cell we biopsy may not be representative of the rest of the embryo. So the diagnosis, now this is a really evolving field at the moment. For many years we did fluorescent in situ hybridization from the early 1990s, and this was a method where we could analyze chromosomes. So we basically had DNA probes that were fluorescently tagged, as shown here in different colours, and you spread the blastomer and had a look at these chromosomes using a fluorescent microscope. But this technique is not that good on single cells for many reasons, and now we've moved on to other techniques. But certainly to look at a specific gene defect that's just affecting a gene, we still use the polymerase chain reaction. The basic technique is amplifying that region around the mutation that we want to look at, but the way that we then analyze that product has really escalated, and there's many, many very high-tech ways that we do that. So arrays CGH and also single nucleotide polymorphism arrays have really come in now in the last few years, and these have now replaced, certainly array CGH is replacing FISH to analyze chromosomes. If we're using the SNP arrays, we can also use something called linkage analysis to look at single gene defects. And sequencing is something that's only appeared on my slide in the last year or so. This is now really almost the holy grail. This will be to actually sequence and read every single base pair, maybe of the whole genome of the embryo, but certainly for now we can do the specific regions we're looking at, which will give us the whole picture. But certainly in the future, we would be able to use this technology to get the genetic sequence of the whole embryo. And then we'd know everything we could know at that time about it. So these are some very early FISH pictures. This is for a Robertsonian translocation, which was between chromosomes 13 and 21. In our lab, we used 2 probes for 21, just as a double check. Obviously, 21 was something we had to be very careful about. So this is a normal cell. There's a 21, a 21, and 2 13s. And this one you'll see is abnormal. There's only 1 21 and 1 13. Now, comparative genomic hybridization is used for array CGH. And what we do first is we take our biopsied cells and we amplify the DNA, and then we label this DNA with a fluorescent compensate, for example here in green, and we label some control DNA in red, and we combine these onto an array, and they're competing, so it's called comparative genomic hybridization. They compete for the spots of DNA that's labeled onto these arrays, and we run these samples out, and it goes into a very complicated bioinformatic algorithm, and it will give us the results. So the sorts of things we see are here. These shaded bars are different chromosomes, so it's got 1, 2, 3, 4, 5, up to 22 X and Y. And all these dots are the clones of DNA for that specific chromosome. Anything in between these lines, if it's just the odd one, it doesn't matter, but all of the clones mainly should fall within these green and red lines. Anything above the line is a trisomy, anything below the line is a monosomy. So here we can see an abnormality in chromosome 10. This is actually a single cell amplified from a trisomy 10 cell line. This is from an embryo where everything was obviously totally crazy. It's actually monosomy for 2, 7 and 9, so there's 2, 7 and 9, and trisomy 6. But when there's so many abnormalities, it's actually got much more noisy. And this is one of the first blastocyst biopsies that we did and used an array. So here was the trifectoderm biopsy that we did that showed a loss of chromosome 22 and that it was a male. And because it was abnormal, we kept the blastocyst and then we did the array CGH on that, and we confirmed that it was a male embryo with a loss of chromosome 22. So blastocyst biopsy and using these arrays is a really neat technique now for doing PGD and PGS and looking at all these chromosomes. We can also use it for the translocations. This one's a 13-14 translocation and is showing an abnormality there in chromosome 14. So I just wanted to share with you a little bit of the data from the ESHRE PGD Consortium. If you go to eshre.com, you can get to the consortium webpage via the special interest group in reproductive genetics. And many of the papers, all of the papers I'm going to tell you about, are freely downloadable from the ESHRE website. So we set this up, as I said, in 1987, and I've been involved with it since that time. And one thing we do is annual data collection from the centres that submit data to us. We collect quite complicated data. It's not from all the centres worldwide, but it certainly gives us a very detailed picture of a few centres. And we publish these reports on the data every year. And a few years ago, I got this data together and we published a 10 years of data collection paper. So it's very readable and tells you about what's happened in the first 10 years. So this is actually first 11 years of data. It just shows you the number of cycles that have been done. So we split the data under PGS, social sexing, sexing for X-linked disease, chromosome abnormalities and monogenic disorders. And we can see that the PGS, the aneuploidy screening, accounts for more than all of the other PGD indications put together. So it's certainly a very large use of the technology. And I unfortunately have to talk to you about misdiagnosis. There's a number of reasons why misdiagnosis can occur in PGD. It can be contamination from sperm, cumulus, DNA in the lab, and cells that are in the lab. The mosaicism I mentioned can cause misdiagnosis. The lab could transfer the wrong embryo. Hopefully that never happens, but in a normal IVF that's not so critical, as long as it's from the same patient. But in PGD, if embryo 2 is the normal one and the embryologist accidentally transfers embryo 1, that would be a real disaster. Confusing the cells that are biopsied with the embryo number is also something that could happen. And here it says unprotected sex. So these couples going through PGD are mainly fertile. When we do the egg collection, we might not collect every single egg from those embryos. And if we don't, every egg from the ovaries, and if we don't collect every egg, it would be ovulated naturally at some point. And if the couple have unprotected sex during that time, around the time of the egg collection, they could get a spontaneous pregnancy. And we're certainly aware of some cases where that's happened. There's been a case recently for us where we were supposed to do a blastocyst transfer. They didn't have any normal embryos to transfer, and then a few days later she told us she was pregnant, and that had to have happened from a natural conception because we didn't transfer any embryos. So we're sure some of the misdiagnoses have been from that. And there's a phenomenon called allele dropout in PCR. I won't go into it now, but that also needs to be factored in. So we wrote a paper a few years ago summarising the causes of misdiagnosis in PGD. So the disadvantages of PGD and PGS, the patients have to go through IVF. So if they have to go through IVF anyway, then that's not a big burden. But if they're a fertile couple, then that's a lot for them to go through. And it is very expensive. The diagnosis is expensive, but also the whole of IVF and PGD is expensive. All of the embryos may be affected. We certainly have that in between 10 and 30% of cases, depending on the indication. As I've mentioned, there have been misdiagnoses, and I would say the success rate is slightly lower than IVF. Certainly in our clinic, we're very aware of that. But if we just look at a very average cycle, so let's imagine we got about 12 eggs collected, 10 fertilised, 8 of them were suitable for biopsy. If it was a blastocyst biopsy, it would probably be less than that. Of those 8, 7 gave us a diagnostic result. 5 of those 7 were abnormal, 2 were normal. Of those 2 normal, 1 was a morphologically excellent embryo, which is great. That gave us a good chance of implantation, but the other was a very poor embryo. So you can see that often we are given the situation where we've only got 1 or 2 really good morphology embryos or blastocysts to transfer. And for PGS, it has to be shown that it increases delivery rates from a randomised controlled trial before we can be sure that this procedure is going to be useful to add on. So the advantages of PGD and PGS. For PGD, an alternative to prenatal diagnosis so that the couple can avoid termination of pregnancy, and they start the pregnancy knowing that the embryo does not carry the disease. For PGS, there's no advantage in my view until it shows improved delivery rates. Some people say, well, we could use this as closure after couples have had a number of failed cycles from IVF. I can see the point of that. There may be couples that have had 1 or 2 trisomy pregnancies and also want to go through some sort of aneuploidy screening. But certainly for the majority of patients, we need data to show that this procedure is really viable. So the ethical dilemmas, we need to really think about these, and these are going to get more compounded in the future. I've already mentioned the Save Your Sibling, or HLA. In the UK, we had a lot of discussions about this. Some people felt very uncomfortable that people were going through all these high-tech procedures to have a child who was going to donate some stem cells to save their sibling. It is done. If there was a matched sibling in the family already, then they would do some bone marrow transplantations or stem cell donations, but it makes some people feel uncomfortable. Sex selection, when we do PGS and when we do any sort of array, we can sex the embryos, so that information really should not be given to the patients because it's illegal in the EU to do that, but it is being done by many clinics. But that leads to a whole imbalance about gender. Is it an abnormality to be a gender of a particular sex? And it gets into a lot of ethical discussions. And in the future now, we've got new technology that will give non-life-threatening. So who decides? So in the UK, as I said, we're governed by the HFEA, but many countries don't have any regulations on this. So with this new technology, we can test for everything. So where's this going to lead us in the future? So for PGD, the new technology will allow us the diagnosis of more diseases. The good thing is that it's making it much simpler to do a diagnosis and much quicker to develop a new test. There's something called whole genome amplification that will randomly amplify the whole genome, and with that product we can then do many things. We've got all these arrays and the sequencing, and we've also got now blastocyst biopsy and vitrification. So vitrification is important for 2 reasons. The first is that it's becoming popular now to biopsy the blastocyst or the cleavage stage embryo, then vitrify the blastocyst, and then take 1 or 2 months to do the diagnosis and transfer any normal embryos back to the woman in a future cycle. And that will make this technology more widely available, certainly globally, but there's maybe evidence to show that that actually helps increase pregnancy rates. But vitrification is also important if we end up in the great situation where we have 2 or 3 normal embryos left over from the couple after the transfer, then obviously we'd vitrify those embryos and have those for a future cycle. So for PGS, as I've said, in my view, we need randomized controlled trials to see if it's a valid procedure with clinical significance. The data at the moment, there was 10 randomized controlled trials on cleavage stage biopsy using FISH, and the view is that FISH is not a reliable enough technique to do this, but we need data on polar body biopsy and trophectoderm biopsy, and I was involved in the initial pilot study with ESHRA looking at a feasibility of using polar body biopsy in array CGH, and now there's a multi-center randomized controlled trial going on supported by ESHRA to have a look at the data on this. We've got all this new technology. We really need to figure out how it all works, and again we can do blastocyst biopsy and vitrification. So just to finish off, a little note about the future. You might want to visit these websites that the 23andMe and DecodeMe use a SNP array to look at multiple sites across your genome, and obviously it would be possible to use these sorts of arrays from a single cell, find out about diabetes, predisposition and many other things. James Watson had his genome sequenced, the whole genome sequenced in 2007, then it cost $1.5 million. Here is a screenshot I did from Illumina a few years ago. It was then $9,500. Anyone could send a sample here and get their genome sequenced. And now it's much cheaper. Now it's a few $100s to get your individual genome sequenced. So we may have a stage where people come to us with their genetic code, and the genetic counselors will be very busy trying to decipher all this and advise the patients what they need to do. But this next generation sequencing has been used in PGD. These were really proof of principle studies to show that it worked. They didn't sequence the whole genome, they just looked at specific areas. But certainly I'd say in 5 or 10 years, it's going to be very I won't say easy, but it will be possible to sequence the whole genome from a single cell. The ESHRE Consortium has written 4 guidelines on PGD, and I recommend those for anyone that wants to learn more, and they're downloadable from the ESHRE website. And looking at the ethical issues, some of us have got together to do this. The European Society of Human Genetics and ESHRE got together. We've written 2 statements over the last few years, and these are quite hefty documents, but they really give an insight into the interface between genetics and reproduction. And this one is looking at some of the new technology that's coming in. Now, PGD is a real collaboration between an IVF team and a PGD team. Our IVF team, this is our PGD team. This is our IVF team at University College and the Centre for Reproductive and Genetic Health. Thank you very much.